Okay, Andrea, go ahead.
Okay, good afternoon, everyone. My name is Andrea Matamoros. I am part of the NUCFIS program, the Erasmus NUCFIS program, and I am performing my thesis master with the medical physics group of the INFN-LNS. So to begin, let me tell you, my talk is about, we call it an overview of Geant4-DNA with Professor Serena Fattori. And how are we going to do this overview of Geant4-DNA? Well, my first task, as I think all the tasks when we're writing a thesis, is to get informed about this. So I am not going to talk like giving a speech about a manual of Geant4-DNA or something like that. No, in fact, I'm going to present through time what has been done with Geant4-DNA in order to understand the capabilities of this toolkit and to understand also what we want to do for my thesis and what we can do and what we would like to do in the future also. So let's go through time. To begin, let's talk about in 2009 when we have the first article. It's called the Geant4-DNA project, basically presenting what is Geant4-DNA. So to begin, as the professor said, we already know that we have a toolkit called Geant4, that is a simulation toolkit using high energy physics for the simulation of particles and matter, right? What the interaction particle matter. But what happens, we have used this simulation toolkit for the macro scale. Like, yeah, we have simulated detectors and other things, other processes, but what happens when we want to simulate DNA damage? First of all, why we would like to simulate DNA damage? Well, we know that one of the principles, is essential targets in our treatments, it's the DNA, right? So what we want, the efficiency of treatments are based basically on how much DNA we break. So in fact, if we break a lot, then the cells will produce, we will have a deficit of the cell, and then it's a good efficiency of the treatment. So for this, can Geant4 simulate this nano scale procedure or phenomena? Well, in this case, that was why the simulation toolkit Geant4-DNA was created. And if you can see, it involves a lot of biology and also chemistry. We will see further that it has a very high component of this. And well, all of it is this. And so for this, excuse my biology. And so what are we talking about? First of all, this one was born in 2001 at the European Space Agency for this, to estimate the biological effects of the ionizing radiation in the explorations. But then after, since 2008, was coordinated by two centers, the INP3 and the CNRS. This one, as Geant4, uses the object-oriented technology, C++. But something that we must know is that the main objective of this Geant4 is to just simulate DNA damage induced by ionizing radiation, but they want to have a simulation platform that is experimentally validated. So we will see after, when I'm talking about the other articles, that they're in fact doing a validation of all these processes. Why we are talking about processes? Well, we know that when we have irradiation, physics processes occur, chemical processes occur. So we have to explain these processes by the simulation. The structure of this physics process in Geant4-DNA, are classified like, we have classes. We said we are using object-oriented technology, right? So we are working with classes. We have process classes that then these ones have a specific model classes in order to obtain a physical quantity. And these ones are all these process classes in this one are a step-by-step tracking. And also these models are redesigned from Geant4 because as we're talking in, as we said before, we are talking about a different scale. So this is the nanoscale and this is low energy limit. So we need to redefine these model classes. As we can see here, these are as I was saying, the process classes. Like each particle that Geant4-DNA can model has their own process classes. Until now, Geant4-DNA can have the projectile electrons, protons, hydrogen, helium ionized one time and helium ionized 2 times. And these ones, each of them has different processes, and the different processes are described with different models. For example, excitation with this model, and each of them has different energy limits. So if a particle is being tracked and then the energy goes below this low energy limit that is described over here, then the particles are stopped and they deposit the energy locally over there in the medium. So this is about the models and the process. Over here, you can see that I mark over here excitation and charge decrease. I think there is a mic on. No, okay. So excitation and charge decrease, for example, they were completely new for Geant4-DNA, completely like they are specifically for Geant4-DNA. And over here we can see an example on what is this step-by-step tracking. This is 1 kilo electron volt electron. And over here we can see that in the whole time of the simulation, we see the 3 processes that the electron can go by. For example, the black spots are elastic scattering, the red spots are ionization, and the purple, I think it's purple pink, is excitation. So each point on it corresponds to a single physical interaction. But what about the modeling? Okay, we know that we are modeling something different from detectors, but now we want to model DNA, we want to model cells. Well, the modeling, yes, we can use the geometry of Geant4, the capabilities of Geant4 to build these geometries. And a specific thing that we have to say is that the modeling, when we do these simulations, they can be voxelized or atomistic. And the one that is for our interest is the atomistic, because we know that we are working with high granularity simulations. So these need to be very specific and consider everything. So the atomistic modeling is the proper one for this. Now, if we continue, we move within time, we go to 2010 and we get to the first report, well, one of the first reports about the comparison. As we said, Geant4-DNA wants to provide a tool, a simulation tool that is compared with experimental data. One thing that we must know over here, if you want to check what is an example of Geant4-DNA, when the classes over there are labeled in this way. First, we have Geant4-DNA, we call it. Then we have the model name, then the process name, and then at the end model. So for example, here in the third row, in the third line, we can see that it says Geant4-DNA born ionization model. So it means we are simulating ionization, the process of ionization with the born model. So when you are reading the codes, if you see this, then you will now understand it. And one thing we must know in this article is that experimental data that was used to compare was acquired in the very low energy in water vapor. Why this is relevant? Because these processes that I mentioned before, they are all based in liquid water. So you must be wondering, okay, but why are we comparing between water vapor and liquid water then? Well, this was considered because in order to build targets of liquid water for this kind of experiments, we need targets that are really thing. So in order, because we are working in low energy domain, so there is a high probability that there won't be transmission of our projectile, so it will get trapped in the target. So for this, the experimental data is acquiring water vapor. And yes, considerations were made in order to compare it with the liquid water model of Geant4-DNA. As you can see here, the results for electron and protons, when we compare the Born ionization model, it has a very good agreement with many of the experimental data over here. So we see that we are making a validated, they are making a validated toolkit. And as we move, we see more articles talking about radiation damage on the DNA. And in this one, there is a big question asking, is nuclear DNA the exclusive radiation target in the cell? Just a little bit for you to see it. Again, excuse my biology. We know that the DNA is in the nucleus of the cell, but also we have the cytoplasm that can be affected by the radiation. So, oh, sorry. So with this simulation, we are trying to see, okay, how much it can be the cytoplasm affected. And why this is important? Okay, this is important because healthy cells generally have a lower nucleus cytoplasm volume ratio than many cancerous cells. This means that for healthy cells, the cytoplasm is generally, not always, a little bit bigger than the nucleus. So, well, the volume ratio, right? And for this, in this simulation, a virtual cell model was created containing the nucleus and the cytoplasm, and the ionization and the energy deposition was evaluated. This one was a simple modeling. So it's a sphere of 5 micrometers that simulates the cytoplasm, material, and then a sphere of 2 micrometers with the nucleus material. And this was all submerged in water. And 3 cases were considered: varying chemical composition, varying density, and varying the nucleus volume. So we want to see first what happens if we consider only water as the material for these 2 components, nucleus and cytoplasm, or if we consider the real chemical composition of these ones. And as you can see here in the table, we have described the percentage of mass fraction for each of them, for the cytoplasm and the nucleus. The second case that was considered is varying the density of the cytoplasm or of the nucleus. And here I'm going to show you the results for the cytoplasm, and also varying the nucleus volume, what happens. So first, considering the chemical composition, you can see here in the results that there is a difference between realistic cytoplasm and water cytoplasm when we evaluate the number of ionizations. So we see that the realistic cytoplasm has more number of ionizations, while in the water, we see that even if it's, in the nucleus, even if we see that it's a water nucleus or a realistic nucleus, they are the same. They have the same behavior. Over here, you can see that they are literally one on top of another one. Over here, you can see a little difference, but not like in the cytoplasm. So, okay, considering realistic, it's important, okay? So for the next one, when we want to evaluate how it affects the density, we see that it's still, this one considers realistic cytoplasm and nucleus. And here we will vary the density of the cytoplasm. So we have this behavior that when we increase the density, we see that the normalized energy deposit over the cytoplasm is still, it's increasing also when we have energies from, well, even from 20 we see that from 20 to 50 initial electron energy, that goes increasing. So we see that the density makes an effect also on the simulation. And to end, we said that we were going to vary the size of the nucleus. So when we vary the size of the nucleus, as you you can see here, the black line simulates the total energy deposit over the whole cell. So these two is the black one represents the adding of these two. And as you can see, when we increase the size of the nucleus, the energy deposit over the cytoplasm goes decreasing, and the one of the nucleus, of course, goes increasing. So what is the importance of this article for simulating DNA? Well, this article basically tells us that when we want to simulate a cell, we indeed have to consider the characteristics of the cell, the specific characteristics, and also the sub-elements like the cytoplasm of the cell, not only the nucleus. So we go continuing, and we have the next report about the Geant4-DNA in 2015. It's a track structure modeling in liquid water. It's a review of Geant4-DNA. And in here, it's important we talk that when we want to simulate a DNA damage, we have to know about the 3 stages of the irradiation. We know that physical stage is when we transfer the energy to the medium, and this one happens around 10 to the minus 15 seconds, around there. Then when we move to 10 to the minus 12, we go to the physical-chemical stage where the radical formation and the dielectric relaxation happen. And then we go to the chemical stage where this one is around 10 to the minus 9 seconds. And these new species that were created before in the physical-chemical stage are now interacting within them or with the ones that already existed. So we see, and we must know that in the chemical stage is where the DNA damage happens. So we see now that chemistry is really important. The chemical stage is really important in what is simulating the DNA. So for this, we must know that Geant4-DNA has constructors that contains all the processes that I mentioned before, the physical processes, the chemical processes. And for this, we can mention the 2 constructors that contain all the processes. That is the Geant4 electromagnetic DNA physics and Geant4 electromagnetic DNA chemistry. So when you see these ones are called in a code or something like that, you must know that, okay, these are already contain all the processes that Geant4-DNA has. In here in this table, you can see the different quantities that we can obtain with these constructors for the different particles. And also in this article is really interesting that at this moment they present all the examples that Geant4-DNA has created. It's several by now, and as you can see here, they are in the standard part of the examples of Geant4. And I marked 2 over here, but also these 3, because I said about the chemical constructor, these 3 are important because they explain exactly how to use these constructors and which quantities you can obtain from them. And here we have the DNA physics and the whole nuclear DNA, which the DNA physics, I will talk about it later, but basically teaches also how to use the constructors of physics. And also the whole nuclear DNA, which I consider it was very interesting. That's why I mark it, because this one says that it was created in order to create simulations by the PDV bank, the protein data bank data. So they just load a PDV file and they simulate the geometry. So it's really interesting. And now we travel to 2017 when this is from the talk. I think this is one of the most important articles, because over here we're talking about the simulation of early DNA damage after the irradiation of a fibroblast cell nucleus using Geant4-DNA. Basically, I'm going to show you because in the previous slides I haven't shown you any model yet, but right now I'm going to show you a simulation that in fact this is what Geant4-DNA is like looking to do, simulate these kind of things. And this is important because also another tool is involved at this moment of simulating the DNA, and it's the DNA fabrics. The DNA fabrics was created by the IRSN and the SimAlgo technologies. This one, basically, it's a tool that it's dedicated to the creation of and visualization of DNA geometries. Is specifically for this. So these simulations that you see on the right, this is created by DNA fabrics. But you're gonna ask me, okay, why are we talking about Geant4-DNA? And now you involve something else like DNA fabrics. Well, the thing is that DNA fabrics, what we simulate with it can be exported to Geant4-DNA. So basically we build the target with DNA fabrics, and then this one is exported to Geant4-DNA. They created their own application to do this transfer. And then Geant4-DNA involves the processes and everything that we want to simulate. So, for example, in this simulation, first of all, it's a fiber. It's an heterochromatin fiber. And what is this? So, first of all, to create this chromatin fiber, a nucleotide is created. As you can see here, it's not only like we build a cylinder and that's the nucleotide. No, we go in a specific. Over here, we consider 5 units to create the nucleotide: the phosphate group, the deoxyribose, the 5 bases, the adenine, guanine, cytosine, and thiamine, and they are all surrounded by 24 molecules of water. As you can see here, they are all spheres, and these ones are connected within them. And you will be wondering, hey, but that looks like they are coated. Yeah, DNA fabric does this in order to avoid overlap within them. And then, so all this one compose a nucleotide. Now this nucleotide, 20 of them twisted in the C direction, they create a bDNA double helix over here. Now this double helix, it's wrapped around a sphere, or this is red sphere over here, that represent a histone, and then this one creates a nucleosome. So this nucleosome, they are connected by linkers, and then these ones are the ones that create the chromatin fiber, as you can see here. In this part, you can see that different configurations of the condensation of the DNA are created. In these cases, voxels, they are called voxels: voxel straight, voxel up, voxel down, voxel right, and voxel left. And then all these voxels are contained into domains, and these domains are the ones that create the chromosomes. So now when we create the chromosome, we can have our cell nucleus, that is this one over here, which is an ellipsoidal with size of micrometers. And each, this one is for humans, so we have all the chromosomes, that is 46, yeah. And this one then, it's after irradiated with protons. I didn't present the results over here, I just wanted to show you this geometry that was created. Why? Because after we will see that the next articles will take this simulation into account. So we will see after it. Before we wanted to study another report about the Geant4-DNA project. Again, we are talking about how is this evolving in the sense of the physics and everything, and the chemistry also. So one thing we must know is that when we have Monte Carlo codes, they can be condensed history or track structure codes. So what does this mean? Well, condensed history codes, they group many interactions, physical interactions together, so they speed up the simulation. But this makes this makes them having a spatial resolution less accurate, and we need a spatial accuracy that is high because we have very small targets, right? And we are working with a high granularity geometries. So for this, we need track structure codes. The track structure codes are a detail. They have a detailed treatment of all interactions because they are based on single scattering models, and also they have an appropriate spatial resolution for the small targets that we are creating. So these track structure codes, okay, we said before that there were constructors. You remember, maybe we said that there was a chemistry and the physics constructor. But this physics constructor has also evolved in these track structure codes. So when you will see, like if you are doing a simulation in Geant4-DNA, that when the physics is called, they can be calling any of these constructors that I list here. There are 3 that are essential. Well, the most important ones, the Geant4-DNA physics option 2, option 4, and option 6. So option 2 is the first set of models, but this one presented deficiencies on the inelastic models. So for this, the option 4 was created. So in order to have better results, so it has an alternative model for the inelastic and also updated cross sections for excitation and ionization. And also we have the option 6. The option 6 is not an upgrade of the previous ones. No, on the other side, the option 6 is an implementation of other Monte Carlo codes called CPA 100 in that they model the physics in a certain way. So these codes were implemented to Geant4-DNA, and that's the option 6. If you remember, I told you that I was gonna mention about the DNA physics example because this one makes a comparison of this, how these constructors that I mentioned before work. So if you can see here, for example, when we talk about protons and hydrogen, in any of the options, you would see exactly the same results. Okay, so when we want to model protons, there is not such a big deal which constructor we choose, but when we want to model electrons, in fact, there is. As you can see here, we said that, for example, the elastic that is the green, we said that there was some problems describing the elastic the elastic process, and this was it was because it was giving more values, overestimating the frequency. This is frequency that happens in the simulation, and over here we see that the value goes lower. Also, we see that the option 2 is modeling other process that the option 4 is not. And also we see a difference in between the ionization and the excitation processes. So depending on what we want to model, you can choose, but option 4 is the one that the other, like I have been checking, the other articles are always choosing option 4. So continuing, like this one also, in 2018, a simulation, well, we are getting into DNA damage. Okay, we already talked about, okay, what is Geant4-DNA? How is it to simulate DNA? But now when we talk about DNA damage, how do we simulate this DNA damage? Well, this article, it's a mechanistic DNA damage simulation in Geant4-DNA. This one is composed by 2 parts. It's really interesting because first they tried it on a simplified geometry, and then when they obtained the results, they decided to try it on a bacterial geometry, on a bacterial cell. in the E. coli, to be specific. And this is, and here I'm gonna explain you more how is we define the DNA damage when we are doing the simulation. So first of all, we must know that there are other codes that are dedicated for this to simulate to model what is DNA damage, like the PARTRAC. And also there are other studies that make their own simulations, like the N97. In fact, in this article, what the purpose of this one was to just create this simplified geometry, but at the same time, compare it with these codes, their results, compare it with the results obtained of DNA damage by these two other codes. So we are validating also Geant4-DNA not only with experimental data, but with other codes. So we see in here that we want to do a comparison, but it doesn't mean that everything has to be the same. In fact, we are focusing on the N97 study, and this one is not it's not the same geometry as you can see here, because in here they just created a sphere of 3 micrometers, and then the source was coming out from this other sphere of 0.5 micrometers, and all this sphere was filled with these rectangles that they have 216 base pairs. So we see that we must know that the other codes does not simulate exactly the same geometry, but what we are comparing, what we want to make it close to it, is the radiation damage classification. Because to define, to measure radiation damage in a simulation, we need to have to define, okay, if this happens, we have a single strand break. If the other thing happens, we have a double strand break, and like this. So this is what it's compared, what is the same as the N97, and this radiation damage depends on complexity and source. So when we talk about complexity, I'm gonna show you better after. When we talk about complexity, we define the DNA damage. This example defines these two parameters, like DSB is the maximum distance between two single strand breaks on opposite sides on the DNA chain for the two nearby breaks to be considered a double strand break. And this one for us was of 10 base pairs. And also another quantity that is really important is the DS, that is the space between two damaged DNA segments for them to be considered independently. In this case, is the length of the DNA segment that we put in the rectangle. That is 216 base pairs. So now we see when we want to simulate something, we have to do a radiation damage classification. Here you can see it better. Okay, this code will detect a single strand break just like this. As you can see here in the left, this is a single strand break. Then 2x single strand break, this is 2 times single strand breaks, is when the distance is higher than the DS that we mentioned before. And as a single strand break plus is this one over here. 2 single strand breaks, it means that we have a higher than the double strand break. They are in opposite sides, but the distance is higher. So we cannot consider it a double strand break, while this one over here is a double strand break. Remember that yes, the DNA can have single strand breaks and double strand breaks, but what we want is a double, it's better if we have a double strand break, because with a single strand break, the DNA, it's more easy to be repaired than with the double strand break, right? If we break it, then we can produce cell death if we have a lot of double strand breaks. So over this, with this, we go classifying the double strand breaks also like this. And also by source, we mean if the single strand break or the double strand break is by indirect or direct or mixed or hybrid source. as we know, the radiation damage can be produced by direct and indirect sources. Direct would be the electron directly to the DNA, and the indirect would be the other species that were created in the physical-chemical stage. So like that, we go classifying by complexity and source. So now we see when we want to model something, we have to have this kind of classification, and these are the results for obtained in this moment. As you can see here, these are the considered values in the modeling in order to the results to be comparable to N97. They call it the best match. And as also this article compares the option 2, option 4, option 6, and also with the N97, how they simulated the physical interactions. So as you can see here, the option, as I said before, the option that it's more selected is the option 4, and you can see here that it's even the one that is closer to the N97 in here, in here, in here, in each of the different classification damage that we have for the single strand break and for the double strand breaks. Also, you can see here the results obtained with respect to the source, well, to respect to everything, comparing it at different energies. Is the Geant4-DNA with the option 4, but at different energies. And we see over here, why they did it like that? Well, they want to know if they are close in the different energy scales. Like you see that over here is 300 electron volts and over here is 4,500 electron volts. So we can see something that it must be a remark from this is that we have direct and indirect damage, right, in the double strand break. And always our Geant4-DNA is this one. Our simulations of Geant4-DNA would describe more indirect than direct damage than what is the N97 over here. So this is, yesterday we were talking with the biologist of the group, and he said that what is important is the amount of double strand break, so it's okay. So, but in fact, they just did this to validate the Geant4-DNA, and in this part they said, okay, it's not agreeing with N97, but in fact it's agreeing with the other simulation that is the part track, so it's good. Then this one was validated with the bacterial cell, and like that in the next years we see, like the last 2 years, we see this simulations that are getting like more realistic. In fact, this one, it's it creates a fractal geometry, and the cell nucleus is all it's more compact. It has a higher density, like more realistic density than the previous simulation. So we see that we are getting into more realistic. So this was 2020, last year. So why we went through all this story about it? Well, now I'm gonna talk about why this is important for me right now that I'm doing my thesis, all these articles and everything. Because to begin, we have, let's make a versus or something like that, of proton therapy versus conventional radiotherapy and carbon therapy. Well, we know radiotherapy does not have a Bragg peak, so that gives more points to what is proton therapy, right? Because we have a more localized energy and we avoid to irradiate normal healthy tissue, right? But what it's when we compare with the carbon therapy, we know that carbon therapy is used for more radio-resistant tumors, right? But we know that the carbon therapy has economical and radiobiological issues. We still have some doubts about the radiobiological effect on the tissue. And so we are thinking, okay, proton therapy is okay, but also we have that protons are just a little bit more biological effective than photons. So how can we enhance the biological effectiveness on this? For this, it was a study that if we use boron 11 into, if we have cells with boron 11 and we irradiate protons, we can trigger a reaction that it's here. It's the proton plus boron 11 producing 3 alpha, a nuclear fusion reaction. And what happens with this one? Well, as you can see here, we have a generation of short range, high LAT alpha particles. And as you can see in the image, well, when we throw protons, we have more isolated damages that, as I said before, these are single strand breaks. While when we have alpha particles and that this reaction is triggered in the Bragg peak. So these are these are getting into the DNA, into the cell directly. So the alpha particles produce more cluster DNA damage. So we see that this can enhance the radiobiological effect, yes. And our team did the first experimental proof of this to enhance the proton therapy effectiveness, but now it's the time to do simulations about this in the DNA scale, what does happen. And for this, my thesis subject right now, it's about Monte Carlo modeling of LAT and RBE in proton therapy at the nanodosimetric scale and in presence of boron 11 radiosensitive isotopes. So for this, we are working right now with an example that is called DNA damage 1. You will now understand why I went through all the articles, because this example is created by it has three components. First, the target is created by DNA fabrics. Then all the processes are implemented by Geant4-DNA. And then the output is a study by root. And this root, it analyzes the energy deposition, the location of the damage, and many other things. And at the same time, it produces an output with a TXT file with an SDD format. The SDD format is the standard DNA damage format created a few years. I think this is very new, the format. I don't remember the year, but when we create any simulation about DNA damage, now the information has to be deployed with the SDD format. So right now we are studying, as you can see here, I just wanted to show you that DNA damage is just Geant4-DNA is just an extension of Geant4. We still have the same classes that we need in a Geant4 simulation: the action initialization, the physics list, the run action, the detector construction, all that. But yes, we vary some things, like the chemical are implemented in other classes, and the Geant4-DNA, the DNA fabrics is loaded with the DNA parser, for example. And this is, I wanted to show you, this is the output. These are the outputs of root. These are the 3 root files that analyze this output and produce the SDD format. And this is the fabrics. It's the fabrics Geant4-DNA. It's the one that exports the target from fabrics to Geant4-DNA. That's what it means, fabrics to Geant4-DNA. This is an example of how it looks, the fabrics, how we generate in fabrics the geometry. As you can see here, it goes detailing each of the components of the DNA line by line, and it goes creating the linkers, the nucleosomes line by line, sorry. This is how it looks when we run it. I wanted to show you this because, as we said, the stages. If you see here in the left side, it says physics stage ends. When the physics stage ends, we know that the chemical stage starts, and it's all this. It goes analyzing all the reactions that we can have over here, and the timing at which each reaction happens all around. So yes, this example is really heavy. In fact, I I've been running it with protons of 5 keV. The default one comes with 200 particles, and it takes like 2 hours in my computer to around there to obtain the results. And this is how the simulation works. In fact, they are simulating 18 nucleosomes, a chromatin fiber. They are simulating a chromatin fiber composed by 18 nucleosomes. And in this way, it's without zoom, so it looks not so good. But over here, we can see each of the nucleosomes, the 18 ones creating the chromatin fiber. Over here, we see better images that Juliana provided to Professor Serena. Thank you so much for them. And here we can see when the irradiation happens, how it looks over here. But right now we are working with this example, and yes, what the next step for my thesis, as we said, we want to model LAT and RBE, and this is the next step of this. Yes, we already analyzed this example, but right now we are analyzing some problems that the example is presenting when I run it or when I am analyzing it. So this example is new. In fact, they say that if there's so debugging problem, that we should tell them. So we are in this phase right now, and also I am shooting at different energies, and from different, now we want to move the irradiation point. Why? Because we also have the SDD format over here. You can see it. This is how, whenever there's a simulation of the DNA damage, this is how the format that the simulation must present it after. And these the last three components over here, you can see that these three integers, each integer represents something: where the damage happened, how the damage happened, the amount of damage, the amount of single strand breaks, the amount of base damage, and the last should be the amount of double strand break. But until now, I haven't found any double strand break, and I think that is the important thing of the simulation right now. So we are wondering some things about this example. We will have a meeting with the developers as soon as possible, and well, this is my work until now. Thank you so much.
Thank you.
I don't know if you have any questions.
Yeah, thank you very much, Andreas. Really, first of all, I want to compliment with you because you really made a lot of work, and it's a really good overview of what we can do with this code, also with the helps of peoples that works in the radiobiology fields. I'm happy that there is Ivan today that can report the status of this. I have no, just a question, Andrea. When you show the physical stage, can you come back a little bit in the at the hand? You know, you have this violet.
Sure.
Yes, exactly. On the right side, I saw that there are the production of free radicals, no?
Yeah.
At different times. So this is directly the product of the hydrolysis reaction. But this is intended as a chemical stage, no? Not free.
Yes, this is the chemical stage.
Okay.
Yes, so I didn't put the reactions because yes, it's more chemical, so I didn't know, like, if you asked me something about it, I wouldn't be, but the articles define the specific reactions that can be explained, and radiolysis was very important since the beginning.
Yeah, in fact.
In fact, that was the evolution that they have said, like, now we can do it better, now we can do it better, and like this.
Okay. And the question is, there is a way, or apart from the format of the file that now is you are producing, there is a way to distinguish between the physics damage, the direct damage, I mean, and the indirect damage directly from the simulation, because in the file there is some indication, no? We are trying to understand this.
Yeah.
And this distinction is made on the basis of energy deposit, you know, the close.
Yes, that's one of the other questions that we have for the developers, because we found out that in the root files over here, this output to root like this output to roots also, well, they have their own trees, right, with the position, with the energy like that, but they are separated in two branches. No, yeah, in two subcomponents. So we were wondering if one is for the physics and one is for the chemical. So we're wondering about that.
Okay.
We think that it must be like that, like this is the physics and this is the chemical.
Okay, okay, no, thank you very much. I have a lot of comments, but it's not the moment. There is someone that want to make some comment on this.
Pablo, can I ask a question?
Sure, sure.
Oh, it's more curiosity. It's not really a real question. Since I am, let's say, I was in the team of the first proof of the proton boron PBCT, proton boron capture therapy, and so on. I am quite curious if there is an intention within your thesis or your work to look at the difference between an external alpha particle beam with a certain dose on the cell, the effect of this, and the internal one. That means when you have a boron in the cell, because Pablo knows very well, we still can't explain in terms of dose. There are orders and orders and orders and orders of magnitude difference. Those which is released. So in fact, the biological part or some biological consideration that is done through the Geant4-DNA is interesting, but I would be interested to understand the difference between external beam and internal. Or does it make sense what I'm saying, or it will be not?
Yeah. Well, first of all, I want to add something that is very important and I forgot to mention, is that yes, we want to simulate in boron, yes, but as I said before, until now there is no simulations on other things, only on water until now. So maybe for a PhD, this would be better analyzed or something. So, but it would be very interesting and cool to do all this all this study about what experimentally you found on this. But until now, we are thinking on honestly how to do it, how to do the boron part since the beginning. So.
Okay, no, thanks. It was really curiosity for me. Definitely, it is complex, so it's time. Yes.
But in fact, Daniele, this is of course one of the reasons why we are starting. Is not the only, but why we are starting to look in a little bit deeper in this Geant4-DNA. One of the reasons, of course, is this one: is to try to understand if we may have some clear or more clear connection between the physics. Also the physics of the DNA damage to the biological response. Everything is it's very complex. So the first step will be to try to insert inside the cell. I hope that we will have the time with Andreas inside the cells, the simulation of the alpha, of the number of the alpha that we expect to have from the reaction, and see how this may affect the single and double strand break. Then to try to understand how this is translated to the biological work and to see the effect also of an external beam, of course, that is quite interesting, as you was saying. This is something that must be done in connection with the biological output. No? One of the things that we want to do is to compare these results with the experimental data of single and double strand break that are taken from our biologist colleague. We are inside a group. There is a project at the European Space Agency and we are participating. And me, I'm coordinating the section from the radiobiology in connection with Monte Carlo. And the reason of this one of the aim of this project is to try to compare Monte Carlo results obtained with Geant4-DNA towards the real measured single and double strand break. In order, I mean, unfortunately, really there is a black hole between the damage to the DNA, the physical and chemical damage to the DNA, that is what we as physicists understand, and the evolution of a cell to something, the repair mechanism or the crosstalk, how the crosstalk between cells is modified and eventually produce some disease. This is something that is completely unknown. It's a black hole that it's very complex to be solved with the today instruments. Also with very huge computer, you are not able to really simulate the answer of the cells with all its complex mechanism, and it's really so step by step. Geant4-DNA is one of the steps, the basics one probably that is more close to physics, but still far from real biology. Also,
yes.
So it's.
No, my question was more from the physics, as you said, exact point of view. If qualitatively, we can see a difference from an external beam and the beam inside, and then based on that, maybe this could be given to the biologists, and they can start to think about what could it be this or just to understand qualitatively.
Sure. It will be something that we want to do once this basic stage of the simulation is ready. So this is okay.
Very interesting.
There are other comments or questions or curiosity from the other people. So no comment. So I want to tell. Okay, the next meeting will be in 2 weeks, and we will discuss about the work of another our student Alma, about the detector, the silicon carbide detector that we are developing, in fact, also in collaboration with Delay for the laser-driven beams. And then in 1 month, we will have also Beatrice Cagni that will discuss the use of some analysis of another proton-boron reaction that this time is made in plasma, but from the detector and diagnostic point of view. So the other 2 meetings will be this one that are already fixed, were already fixed. So Alma and Beatrice. Okay, may I ask to switch on the camera so we can make a photo and then we will say goodbye to everyone. So
I'm sorry, my camera doesn't work.
Okay, I will put your photo.
Photoshop.
Photoshop.
Excuse me. Hi, everyone.
My camera doesn't work as well.
Can you hear me?
Yes, yes.
Okay. Can I ask some question about presentation?
Sure.
First of all, thanks for your valuable presentation. I have some question about some slides. For example, in damage 1 example, the energy of proton was considered 5 kilo-electron-volts. Yeah. Is there any special reason we know this energy? I mean, in this energy, we haven't Bragg peak. Yes. Why we use this energy for proton?
In here, sorry. Yes. In here.
Damage 1 example.
Yes.
Was energy of proton 5 kilometers.
Yeah. In fact,
why we consider this energy for proton?
Okay, this is the default example. This is the default parameters of the example. Right now, I'm throwing different energies at the same amount of protons. But this is just default. We can change it, and that's the thing that we want to find. When do we have the Bragg peak into our DNA that we produce the double strand breaks?
In this energy, we have Bragg peak in DNA.
No.
Have
No.
the Bragg peak in this energy of proton?
Well,
no, may I comment, perhaps?
yes, because I don't understand the question.
No, I think, okay, so this one is the default, I think, that no, Andrea,
Yeah.
you used. By the way, regarding the energy, no, of course, this is not the real energy that we are considering in the simulation. No, the proton beam energy, it will depend on the position mainly of the tumor and where we will consider the position of the boron 11, no? Because we want to study the boron 11 and the interaction of the boron 11 with the proton. The proton, 5 keV, it's a very low energy proton. I mean, it's the proton that is stopping basically in water. So it's not in the Bragg peak, it's after the Bragg peak in this case. So if this is the question. So when we will go to simulate the real situation, what we will do, we will have a real beam, that is the clinical beam that we use. We shoot this beam. This beam has different components, from 30 MeV up to, it's possible, let's say, 12, 10, 5 MeV. But this beam starts to stop in water and may reach also this very low energy condition, but there will be also, of course, protons that will have an higher energy. So this is not the real condition. This is some study condition that we are considering.
Yeah, right now we are just literally just playing and understanding the example right now. And then we will do it more specific.
Thank you. Thank you for your explanation.
Okay, so I try to make some photo, but can you switch again the camera, because I'm not sure. Oh, hi Alexandra. Ciao. I saw you. Andrea, please switch on the camera. So no, 2 volte. Where is Andrea?
Is that I have my 2 computers connected, sorry.
Okay, so double Andrea.
Yeah.
Okay, at the moment that I make, so cheers. Ok.
Il mio sorriso è il più bello di tutti.
Meraviglioso. Okay, so thank you very much. We will upload the slide and the video as well if Andrea allow for this. So we will upload the video. And okay, so thank you to everyone. Have a lovely weekend, and we will meet soon. Okay, ciao.
Bye.
Ciao. Bye.
Ciao.
Bye.
Ciao.
Bye.